so that one is easier. All right, the phylum Nematoda. What animal falls under that? Nematodes, yeah. So we hardly ever see them, but they are by biomass the most abundant animals on Earth. Platyhelminthes. Yeah, the flatworms that kind of look like an arrow. Their key trait, what kind of came along with them, was this idea of cephalization, kind of the nervous system kind of starting to join up towards one area of the body, forming what would eventually become a brain. Yeah. And then what we saw in 1102, if you kind of were able to get a little lucky and find this one's brain, that Annelida, what animals are these? Yeah, earthworms. What's that? The earthworms, right? So we also dissected one of the next ones, mollusca. What animals fall under here? Actually, a wide variety of them.
Snails.
Yeah, snails. What else? Clams. Yeah, and even the octopus and squids are themselves mollusks. Their shells are internal. Then we're kind of getting closer and closer to chordates from here. But actually, I'll kind of skip this one and go to arthropods. What do these include? Spiders, insects, crustaceans too. So crabs and lobsters and crayfish, all the yummy stuff. And then now I'll come back here. I come back to this last because these, in terms of phyla, are our closest relatives, the Echinodermata. And what do these include?
Starfish and mantises.
Yeah, so the sea stars, sea urchins, sand dollars.
Is it the anemone that's the spiky one or
no? No, that's a sea urchin. Yeah.
Mm-hmm.
So that would be Pukumuku to Pokemon fans. But yeah, so all of these are invertebrates. They're not chordates. What we'll talk about today are animals with a true backbone, which is used to define vertebrates, and we'll pretty much just focus on vertebrates. But I do like bringing up that the term for a vertebrate is not the exact same as a chordate. So we'll look at what a chordate actually is. We kind of went over this already.
Is this on Blackboard?
What's that? Yeah, these slides are on Blackboard. What do vertebrates have? They are all bilaterally symmetrical. They have body segmentation. We'll talk about these pharyngeal pouches. So there are a lot of traits that vertebrates have, but there are four derived traits that make chordates unique to all the other animals. So essentially what that means is 4 traits that are unique to them. So they didn't appear until after, until chordates themselves evolved. And those are these 4 things. It's called a notochord, a dorsal hollow nerve cord, muscular postanal tail, and pharyngeal clefts, otherwise known as pharyngeal gills. Now, we probably know that there are other animals that have gills. There are other animals with tails, but there are actually other animals with nerve cords in and of itself. So if you remember from 1102, crayfish have tails. Crayfish have gills. We identified those, but they're not. That's why there are these more technical terms applied to chordates. They have pharyngeal clefts, basically gills forming in their neck region. For crayfish, gills are very different. They evolved, would we say, by divergent evolution or convergent evolution? If they independently evolved a gill-like structure. Convergent. Yeah. So
they're not
crayfish.
related.
Yeah. So they independently evolved gills, but they originated independently. The same goes for the tail. The tail structures of some insects and some crustaceans, like a crawfish or a lobster, is very different than the tail that we associate with many mammals, for example, and fish even. So if you remember from 1102 just now, if you were in my section, but I think everyone has now seen the crayfish, the tail is not a post-anal tail. So if you kind of followed, you would see that the intestine runs the whole length of the tail, and the tail ends at that uropod. That's where it actually poops from the end of its tail. Something that we should know at least mammals don't do, right? If you have a pet dog or cat, you know where its poop comes out of. It comes out before that tail. So it has a post-anal tail, the tail coming after the anus. Now, the notochord and the dorsal hollow nerve cord. Other organisms have nervous systems and a nerve cord in and of itself, but ours is a little bit different in that it's hollow inside. And so it has a different structure in chordates. The fact that it's this hollow nerve cord, what would eventually become the spinal cord. And then this last one, the notochord. Now, when we think of chordates, often the term we think of are vertebrates, right? But not all chordates have a vertebral backbone, a backbone like when you think of a human's backbone or any mammal's backbone with individual vertebrae. Some chordates have a non-vertebral backbone, but it's still, a rigid structure that is similar to the backbone that most mammals have. So we just refer to it as that notochord. So these are essentially those four things visualized: those pharyngeal slits, the gills, the post-anal tail, the fact that the tail goes after the anus, the notochord, what in vertebrates is going to become that vertebral backbone, and the dorsal hollow nerve cord, so what will essentially become the spinal cord. So what ends up happening actually as we kind of progress and get closer and closer to mammals is this notochord will kind of envelop the nerve cord. Because when we think about human anatomies, our spinal cord is actually within our backbone, right? So it's kind of this further protection we get to protect our spinal cord, that nerve cord. The notochord kind of just envelops it and ends up becoming a vertebrae. So this is just a fake animal. But if you were to see the image of this animal, you'd be able to identify those four main features, the ones boxed here, that are unique, that characterize chordates. There are other things, right? There's musculature, the mouth, the brain, but four of these are just unique to chordates specifically. So definitely want to be familiar with these four, especially their specific terms. So the notochord and the dorsal hollow nerve cord, this is the one that students tend to have the most trouble with, because when they're trying to remember the four, they're both chords in some way and a little bit similar, but keep in mind that one of these is eventually going to become the backbone itself, the skeletal structure, and one of these becomes the spinal cord. So spinal cord and the backbone themselves are different traits. And two of the four traits that characterize chordates. Usually the tail and gills everyone gets right away, but one of these tends to get missed by students. What's the sitch? When we describe these four traits, these aren't traits that, necessarily some chordates don't have at all. And this can be surprising to some when we think of humans, right? We're chordates, we're vertebrates. We don't have a post-anal tail, we don't have pharyngeal slits, but at some point in our life we actually do have both of these. So when we compare a chick embryo to a human embryo, early in development we actually do indeed have those gill slits. We just lose them before we come out. And the same goes for the post-anal tail. And in fact, some people actually still have a vestigial tail that is most often removed upon birth, but there might be a little nub there. We kind of lost the purpose for that tail. Tails are good for providing stability for quadrupeds, animals that walk on all fours. Once you become bipedal, you don't really need it as much. Although there are always exceptions. But in monkeys that are mostly bipedal, their tails serve other functions too. They're almost opposable digits. And so these 4 traits we do see in all vertebrate embryos at some point. It's just some individuals retain those after birth and others don't. So this is the point where we kind of talk about the vertebrates. So there are a few animals out there that are chordates but are not vertebrates. I just, will show you just for curiosity. You don't really have to, necessarily know this for the exam, but I just like showing this because the animal you might be thinking of really, you would not associate with a chordate. One of those, kind of the main one, are the tunicates.
What is that?
Yeah, so this is a chordate. It's got all 4 of those features that we associate with most vertebrates. But, they don't have a backbone, obviously. But they are chordates. We would think of these as kind of more closely related to sponges, maybe.
That looks more like a plant than an animal.
Yeah, exactly. But it's actually, we're more closely related to this than any insect, other things with eyes. It kind of just developed a strategy that kind of mimicked that of the sponge, but it is a chordate. In fact, one of them is really cool. It's called a siphonophore. It just looks like these kind of live in the deep sea, and they'll often just look like a floating spinal system, like a nervous system. So these things can get, hundreds of meters long, one individual. And so basically there are these tunicates that kind of grow on one communal branch, and they're all clones of each other. So these are one of those organisms that we kind of, they're kind of in this gray area between, is it one organism, is it multiple? They're all clones of each other, but kind of separate entities connected by one string, essentially, in the ocean. So they can get really long, as you can see. But yeah, these are technically chordates. So even within chordata, within chordates, there's a lot of diversity as to what we'll find. But for the sake of this course, we almost use the term chordata and vertebrate interchangeably because those animals, another example is the hagfish, is another example of a fish-like creature that has a notochord, a dorsal hollow nerve cord, pharyngeal slits, and the postanal tail, but it doesn't have a vertebra. It doesn't have a backbone. And it looks really gross. So this is another one of those non-vertebrate chordates. So when you're thinking of this, note that all vertebrates are chordates, but not all chordates are vertebrates. The term vertebrate is within that phylum Chordata. But yeah, the vertebra is what most of these have. Okay, so before we go any further, we can kind of start the first and really only activity today, which is going to kind of be y'all's attempt at getting through the chordate phylogeny. So one way I like to kind of depict what we've done all throughout modules 3 and 4 is, we've kind of been putting, just like when we think of a microscope, you can kind of increase that lens magnification. We're kind of doing that closer and closer to us, humans. So we started module 3 with just kind of the origins and, the first life forms. And we kind of looked at, didn't even really mention archaea, But we talked about bacteria, and these are both prokaryotes. And then we talked about, we mentioned the eukarya. And so first we looked at it just in terms of this, right? Prokaryotes versus eukaryotes. Now, what we did next was kind of start focusing in. We kind of zoomed in to the eukarya, and we just like put a first magnifying glass into this part of the phylogeny. And we looked at all the diversity within eukaryotes. So one way we can look at that is as that phylogeny, where protists are, yes, that junk drawer, but within all this branch of eukarya, we do have, branches of protists that end up leading to the plants, the fungi, and the animals. And we took kind of pit stops along the way. We looked at fungi, we looked at plants, and we looked at animals. Just a quick refresher. What group do plants come from? Yeah, charophytes, which are within the... yeah, and the common name would be, just so in case you're having trouble remembering, the green algae, right? So yeah, those are the three key words you want to think about though. Charophyte is really that specific member within the Archaeplastidae, which are also known as the green algae. And it's literally in the name of Archaeplastidae, so if you kind of break it up, archae meaning kind of ancient or old, plastid, one of those organelles that we associate with plants specifically, those chloroplasts. So it's kind of the predecessor. Fungi, where do they come from? So I think I heard someone say it. Yeah, Uniconta. We don't spend much time on here, but you want to at least be familiar with that group. So we know where this whole kingdom came from. And then lastly, the animals. Don't necessarily have a supergroup name for them because they're really peculiar protists, but we talked about them on Monday. The name rhymes with Droanoflagellants.
Coanoflagellants.
Coanoflagellants. Yeah, so the Coanoflagellants. And so you'll want to be familiar with, in terms of protists and the importance of the protists, which were kind of everything else that aren't these three, you want to be familiar with the endosymbiotic theory because that's where protists really come in. As those first eukaryotic cells, we kind of get into endosymbiotic theory, the formation of the integration of mitochondria and chloroplasts into what becomes the eukaryotic cell. But then the other thing you want to know is the predecessors of each of these, as well as the hints that lead to us drawing these conclusions. You don't have to worry about it as much for the fungi because we don't really talk about why we know fungi came from Uniconta, but we did spend a bit of time talking about the clues, the traits that plants and charophytes both share, as well as the clues that lead us to believe that the first animals were derived from coanoflagellants. That involves looking into sponges, how they have cells that are essentially just coanoflagellants. They're called choanocyte cells. So you'll want to be familiar with that. So we kind of did that throughout Module 4, looking at each of these groups. And now what we've done, what we did on Monday, was kind of zoom in further into the animals themselves. And we had another phylogeny from there. and we looked at all the different phyla of animals. From sponges, SpongeBob, to jellyfish, and I mean others, nematodes, arthropods, echinoderms, our closest relatives in terms of phyla, and then lastly, the chordates. And so what we'll do today is put one last magnifying glass over the chordates themselves. Why do we do this? Are chordates more complex or superior to arthropods or jellyfish or sponges?
Yeah.
Yeah?
No?
Well, part of it is we're biased, right? How do we kind of measure superiority? In the measure of evolution, it's which is the most successful strategy. Well, all of these are extant species, meaning they're not extinct. They still exist to this present day. Many species outnumber us by orders of magnitude, whether we're talking about bacteria because they're super tiny, or arthropods, which outnumber us, like ants outnumber us by about 1,000,000 to 1. So one of the reasons we kind of keep focusing on phyla and groups closer to us is simply because we're biased, right? We're biased to understand ultimately how we behave, what makes us function. And so the closer we get to us, looking at those organisms can help us understand our own past, right? So we just have a bias to understand chordata. It's not that chordates are more diverse than any one of these groups. You know, in terms of diversity, arthropods are far more diverse than chordates. So just something to keep in mind as we go through this is it's not like there's some superiority in terms of as we get closer and closer to humans. We just have a vested interest in understanding our closest neighbors because we want to understand how we function, right? So
it's not that
that's part of why we do that.
It's not actually that they're superior than any other it's just that like we just have a bias towards them.
Right. I mean, part of it is, you know, how do you measure superiority?
Right.
If it's by number, we're losing by a lot. If it's by, you know, ability to live in different environments, there are far more tolerable, you know, tardigrades probably win that battle because they can even live in deep space, right? So, you know, that the superiority, basically whenever we define an organism as higher order, sometimes these terms get thrown in like more complex, higher order, more intelligent. We're just really saying each of those definitions in our head is, okay, which one of these is most like us? It's not an objective measure, right? Oh yeah, so I was gonna give you all this. What we'll do is this activity is gonna kind of be the last magnifying glass here. So looking just at those chordates. And what you'll want to do is essentially kind of pinpoint the traits associated with each of these classes, and you'll want to complete the section on the back. Once you complete the phylogeny, the section on the back is pretty easy. Just keep that in mind. It shouldn't take too much time. Have jaws. They kind of just will suction on to, they're kind of parasitic. They'll suction on to whatever host they want to feed off of. And as we go through these, I give Pokemon examples. This would be the equivalent of an electric in Pokemon. They don't have jaws yet. They just kind of will suction onto their prey. This is really a hybrid between a lamprey and an eel, but it's the closest thing. So what is that first trait then that evolves that we can put here? Something that specifically separates sharks and rays from lampreys. Or it separates lampreys from everything else. Yeah, the jaw. Yeah, so the jaws and what's called a mineralized skeleton. And actually this use of the word bony, I don't really like that. We can just, really it's better to call it a mineralized skeleton. Basically it's the start of the skeleton as we know it, what we refer to as an ossified skeleton, what kind of everything else has. So the sharks, we can kind of put jaws into, Peter will wake up, we put E here for jaws. And so, we essentially end up with a creature now that, has those 4 hallmark traits of chordata. It has a vertebral column, and then it starts to develop, a jaw. In fact, the jaw is essentially the first part of that skeleton that becomes ossified. So whenever sharks, fossilized sharks are found, often the only thing you find are the bones of its jaw, actually, because that's the part that's ossified. The rest of it is mineralized. It's hard. It gives the shark structure, but it doesn't tend to stand the test of time. And one of the ways we believe the jaw to have evolved kind of follows on this idea that was being suggested with the Cambrian explosion. What was one kind of interaction that's thought to have led to all the species that came about?
Oxygenation.
Oxygenation, but something too about animals. Yeah, predator-prey interactions. Once you have something that can start eating something else, then you get this arms race, evolution for other traits. And predation is one of the strongest naturally selective pressures there could be, because it's literally taking individuals out of the equation at the expense of another individual. So it benefits one species, one individual, at the expense of another. And so that possibly explains why the jaw was the first thing to become ossified. You can imagine animals at this point are already eating other individuals, but some individuals have developed armor, and so the next sort of innovation that evolves is an even stronger bite. So that comes in the form of a stronger jaw that can kind of eat through tougher prey. And we actually think it evolved from those pharyngeal gill slits. So the pharyngeal gill slits have some form of rigidity to them, and so it's believed that those just became more and more rigid until it just formed a jaw bone specifically. And so once you have predation, then it starts, just like with the Cambrian explosion, that arms race. Well, the ones who are going to kind of respond the best to a shark that's constantly eating everything are going to be ones with stronger and stronger armor. So an ossified jaw selects and eventually leads to the evolution of an ossified everything else. In the Pokemon world, Sharpedo is kind of the best example of this because it is a shark that's essentially just a mouth. So the evolution of those jaws in Sharpedo. Okay, so what would come next then? If we're keeping on the activity sheet, looking at kind of the next member in the line. Which one? Lungs? Everyone agree with that?
No.
No? Yeah, rod-shaped bones. So just like this predator-prey interaction, strong predation pressure leads to a strong response as a result. Basically, the sharks kind of take out a huge swath of the population that don't have strong enough bones, and those with stronger and stronger bones start to be more and more represented, and you start to see them start to evolve stronger and stronger skeletons. So the rod-shaped bones. So, sharks do have skeletons in some way, but specifically, these bones are starting to be ossified is the key word. referring to rod shape, but you might see it as ossified, which I like better, especially if you speak any Latin language. Ossified just meaning coming from wissa for bone. So a bona fide bone is what it's leading to. So just like a Sharpedo is now going to start eating a bunch of things, it's going to result in fish that can kind of handle the shark's bite. Stronger and stronger skeletons for more and more protection, and that's where the Magikarp kind of comes in. Magikarps are notoriously difficult to eat because of their hard exterior, and so they're kind of a nice Pokemon to stick here. They're strong skeleton, possibly evolving in response to the Sharpedo's strong jaws. Then, Abraham, what comes next? Lungs, yeah. So yeah, lungs are next. This is the one that, because there aren't that many animals that just kind of are in between the fish and the amphibian, which we'll talk about next. This really only applies to a handful of animals, like the coelacanths and the lungfish. They essentially are these sort of prehistoric stepping stones that lead to a life away from the sea and away from the ocean. But part of that is the development of lungs or lung derivatives, the swim bladder specifically. So lots of fish. Actually, what kind of evolved first was potentially the lung itself. So this ability to breathe outside air. And then some fish evolved a swim bladder in response to that, actually. The swim bladder is a modified lung and not the other way around, actually. The swim bladder helps the fish kind of go up and down the water column. It gives them buoyancy. That is, I'm guessing, going to be a relicant in response. Yeah, Lori.
Professor, can you go back?
Yeah.
What is that Pokemon that's at the top?
Oh, that's a Staryu.
The one below that?
An electric. It's kind of like a lamprey. So what's kind of being highlighted here is the fact that it doesn't have a jaw. So the first vertebrates were jawless, like the electric. The real animal would be the lamprey, which looks terrifying. It's got teeth, but they just kind of, what it'll do is it actually makes a suction, but it's worse because it'll dig those teeth in and then make like a 3-degree turn. So it's kind of like 1,000 daggers and the twist of the knife. And then, yeah, they kind of latch on to large mammals, actually, whales, so aquatic animals, whales, dugongs, manatees, they'll feed off of them. But yeah, these are, this is like our grandfather.
Have you seen that Hollywood, I'm sure all of you, it's like the King Kong movie?
King Kong itself? Yeah.
The movie.
Which one?
The one
Yeah.
with Jack Black.
Oh, yeah, that one.
With Jack Black? Oh, yeah.
Okay. Yeah,
Of course,
so
Peter
there's like a
Jackson.
lamprey, and there's like this huge lamprey that kills a guy.
Oh, I didn't see
You
that.
haven't seen it?
No.
Okay, so it supposedly was like, in the movie, it was like a lamprey that was like prehistoric, still living, but it was huge and humongous, and it killed the man.
Yeah, so these are not big at all. Lampreys are
I
like,
know, they're small. I've
yeah,
seen it,
yeah, small.
But I thought that was pretty disgusting. 'Cause I'm
Yeah,
like,
yeah. I mean, a lot of these kind of alien-looking creatures are the inspiration for most of the monsters we think of. So the xenomorph from Aliens is actually modeled after a couple different species of aquatic invertebrates, damselfly and dragonfly larvae being one of them. So dragonfly larvae, they live in the rivers. They're not the dragonflies you think of. They're just aquatic larvae, and one thing they do is they have this projectile that comes out of their mouth. They don't have jaws, they have arms that developed into a projectile mouth, and that's how they catch their prey. They essentially live their lives like this, and they'll kind of snap prey up and then bring it back to their mouth, just like the xenomorph in Alien kind of has that coming out. So yeah, inspiration for Hollywood horror, another good reason to study all the peculiar animals that are out there. So yeah, lungfish. There aren't that many good examples, but there are fish out there that will actually jump out of the water and they'll kind of gulp air into their lungs and go back into the water, and just a kind of quick way to get oxygen. But it's believed that this was kind of that necessary step to get us to the evolution of the lung. So yeah, this would be the coelacanth would be the other example here. A lobefin or a lungfish here. So what would be next then? We've got 3 more traits. Let's look at our activity here. And so now this is essentially separating the out of the water animals from those aquatic animals, amphibians, reptiles, mammals. They all spend a considerable part of their life out of the water. They don't all live 100% out of the water, but they're mostly independent from the water. So what trait then might they have evolved here? Not quite yet. So they're not fully independent of the water.
Hair,
What?
hair?
Do amphibians have hair?
Limbs.
Limbs, right? So one thing they all have, it's kind of tricky seeing that in a snake, right? That's probably an inappropriate picture for your textbook to have on here. This image is just from your textbook.
Digits?
But yeah, it's because snakes lost their limbs. So really should have lizards on here. Yeah, so snakes did have, well, they evolved from an ancestor that was limbed. They had limbs. You can actually see kind of individuals that look pretty close to that.
So we could
There's like a lizard that looks kind of like a snake head.
Yeah, I mean, there's the skink, which is very long.
So we could evolve in lizard limbs?
You could. You keep asking questions like that? No. I mean, not anytime soon. There is evidence, though, that, I think this has been debunked now, but there was evidence that we might be losing our pinky toes, right? So, yeah, so take a look at that. Yeah. Just because of a lack of reliance for it anymore.
So we're gonna have more tough.
You will. I'm not some bugs. Yeah, maybe. There's one too. It's too much of a tangent.
We're
I'll
eventually
mention
gonna
it
evolve
later.
into Aquaman, like
Yeah. The other animal, this is an amphibian, but it's kind of going almost the route of the snake. It's an amphibious version of this. They still have their limbs, but you can see they're super highly reduced, and they have this long slithering body as a result.
That's cute.
Yeah.
Oh, yeah.
It was disgusting.
There's a Hox gene with that one, right, Professor?
Yeah, so these are close relatives to those oxalotls, which oxalotls also have kind of defunct, well, not necessarily defunct, it helps them. They've had mutations to their Hox genes to the point where they actually are what are called pedomorphs. What's that? This one? Yeah. The San Diego Zoo.
They look so fake.
What's that?
They look so fake. Like
Yeah, no, they're super real though, and I mean you can buy them. The Wooper from the Pokemon is modeled after the Oxalotl.
Really?
Yeah. Oxalotls are what are called pedomorphs. Most newts and salamanders actually look like this at one point in their development, but they end up becoming, they complete their song. When they're done, they're at their zenith. Their pinnacle. The pinnacle of their existence. Yeah, can't you see? No limbs. But they end up becoming, the salamanders that we think of. But all salamanders look like this, but the oxalotl kind of pauses here due to mutations in those Hox genes that don't allow them to develop any further.
And how is that advantageous for their survival?
For them, well, I mean, it's just a sort of peculiarity. they never leave the aquatic environment. Salamanders and newts typically do. So they exist in an environment where they never really had to leave. There's not very much predation they experience. And so a life in the water just kind of made sense for them. You can think that, predation is one kind of driver to get out of the water, right? If you have aquatic predators after you. But it costs a lot of energy. You have to keep developing. You have to lose those gills, form new organs that allow you to live out of water. So without predation, it's an energy it's saving energy to kind of just pause there. So, so limbs though. All these things have limbs, even though that picture is misleading here with the reptiles. Limbs allow for them to essentially, once they're on the land, move around, right? It's pretty useless if you're at this lungfish position, if you get out of the water and kind of can't move from there. So you need limbs to navigate the outside world. But amphibians are still dependent on the aquatic environment at this point. They're dependent on it for mating purposes specifically. So, we got from fins to limbs. A little information there on the amphibious life cycle. So the life cycle of a frog can be applied to salamanders as well. But even though they can live their adult lives outside of the water, they always have to come back to fertilize. And that's because their eggs don't have any way of retaining water. Once these eggs kind of get out of the water, they dry up and are inviable anymore. So they need that aquatic environment to develop into the adults that can leave the water. And so they're still tied to the aquatic environment. So you can imagine a polytoad that can go far into the land, but at some point in its life, it's going to have to come back. And so if we're talking about all the ends of the earth animals can occupy at this point, we're going from a purely aquatic living organism to organisms that perhaps start testing what a non-aquatic life is like, so the development of those lungfish that can gulp air outside of the water, to an ability to actually navigate those surroundings. But they still have to come back to the water. So you can't go too far, right? Amphibians, we hardly find them in deserts far away from water. There are, of course, exceptions where we do see them because there's some aquifer or some oasis nearby, but they can't navigate all the ends of the earth just yet. We need to separate ourselves from water in that instance. So, what evolves next? What's the next adaptation to arise that first appears in reptiles? That's where the amniotic egg essentially comes in. So, when we think of eggs up until this point, think of caviar. Frog eggs and salamander eggs are pretty similar. They're very soft eggs. Once we get to reptiles, birds, and mammals, that egg starts to harden. One effect of that hardening is that the liquid environment within that egg doesn't escape. So it can't just be dried up as easily as it can for amphibians and fishes. So the amniotic egg is that trait that separates the animals from that dependence on water. So I want y'all to remember that. If there's one trait that kind of cuts the tie from a dependence on water, it's that amniotic egg. Reptiles develop other traits too, like keratinized scales and things like that, but the amniotic egg really is that big one. This I won't even mention now. We'll talk about this in systems biology, how frogs breathe through their skin. But yeah, this is just driving that point home that the amniotic egg allows the embryo to develop on land because it's basically a mini aquatic environment. We'll kind of skip that part. Don't worry about this is just reiterating those points that yes, reptiles have scales. They can now breathe entirely through their lungs instead of relying on skin, which is what amphibians rely on, but more of that in Module 5. The amniotic egg really is that important one. And so we go from a polytoad to a septile. You know, a septile now can carry, the females can carry eggs on their back and not rely on that aquatic environment at all because they have these hard amniotic eggs as a result. And so that leads to the last trait and the last option available. Okay, here. Which is for the mammals. And all that's left here is hair at this point. So, you know, hair really seems like, you know, all of these traits sound super important. And so hair is like, okay, what? But think about what hair provides most animals beyond ourselves, right? We've replaced hair now with clothes, but hair allows mammals to kind of even further diversify the environments that they can inhabit. That hair can insulate them and kind of make them kind of, with the hair comes this endothermic ability, warm-bloodedness that mammals have. So we kind of cap things off. So mammals, again, have a lot of other traits. Hair is really just one of them. So mammary glands, the definition of a mammal refers to the mammary glands, the fact that they have organs that can help them produce milk. They also have fat layers under the skin, and because of their warm-bloodedness, what we call endothermy, they have a high metabolic rate. But yeah, that leads us to a point that you want to make sure you're aware of, this difference between endothermy and ectothermy. Endothermy is warm-blooded, ectothermy is cold-blooded. Endothermy and ectothermy are kind of better terms for it, because ectotherms mean that their body temperature changes in response. Ectothermy meaning outside temperature. The outside temperature is what regulates their body temperature. It's why you see lizards, when it's cold outside and sunny, they go out to a rock and start doing push-ups. They're doing everything to warm up their blood. Basking gives them that energy to speed up. Once it's cold outside, they are quickly much slower in speed and metabolism. They slow down everything. Endotherms escape from this in some way. They now rely on just internal mechanisms to keep the temperature for us at 98.6. We don't have to go out every 45 minutes and do push-ups in the sun.
Professor, isn't there like an alligator or crocodile that can dictate its offspring sex based on?
There are a lot of animals that can rely on, so it's environmental sex determination versus genetic sex determination. So yeah, alligators and turtles as well, and other organisms, some insects. The way sex is determined is by temperature. Basically, to produce females, a whole clutch of females, they'll up the temperature of their clutch and produce all females. And then ones that are colder will develop into males.
Whoa, I thought it was going to be like reverse.
Oh, no, you're right, it is reversed.
Oh, okay.
Males are when it's warmer, females are when it's colder. Yeah.
So basically, there's like a community of alligators. They would just decide what they want to be born.
Well, sometimes there is some behavior that actually goes into the selection of what sex your offspring is going to be. Animals have a whole slew of mechanisms to try and divert resources to producing females versus males. The thing is, the population almost always ends up being about 50-50, except for a couple of unique examples. Alligators and crocodiles, that difference in temperature is very easy for them to manipulate, and it's a very kind of specific temperature. So in most alligator populations, it's not like they have to go far away and find a cold spot to produce females. Really, they can just kind of, produce a kind of, lay the eggs kind of in one part of a stream or a lake versus the other. That small difference in temperature is enough to produce males versus females. And sometimes it'll be so precise that the outside clutch of eggs produces females, and the inside clutch produces males because they're being warmed up by the outside.
That's trippy.
Yeah. So there's no genetics in that. Yeah, and then it gets weirder. Some rely on both. But yeah, ectothermy, endothermy, cold-blooded, warm-blooded. And so the last thing, we saw this in the IRAT with the mammals. We can break those up into, you want to know, those monotremes, marsupials, and the placentals, or the eutherians, which is the majority of animals. One marsupial here in the New World, all the rest are exclusively native to Australia. Anyone tell me what the marsupial is here in North America that's native here? Possums, yeah.
Opossums.
Yeah. Oh, did you not say possum?
No. Well, there's both. There's possums and there's opossums.
Yeah. So.
And I think they're cousins.
They're actually they're
Virginia
not
opossum.
scientifically different.
Oh, they're the same.
Yeah.
look
So
different.
there is kind of a difference in terms of one comes from Australia, one from the U.S., but genetically, actually, some opossums are less related to each other than the New World possum. It's a very strange, because even though they're all in Australia, what happened was after Australia kind of separated from everywhere else, that's where the marsupials were, and eventually one of those marsupials made it to the Americas. So
The
it's
Virginia, right? They call
the
it
Virginia possum, yeah. Beautiful
So,
animal.
why do they look different than possums? Why do opossums look different than possums?
Well, think about the difference in environment they're experiencing. So yeah, opossums, I remember in New Zealand, they look totally different. And actually, a lot of clothes are made out of opossum because they're invasive, and they look very different, right? But part of that has to do with experiencing very different environments over a long enough period of time.
Because I used to raise possums, opossums, the Virginia opossums.
Yeah.
And they looked like that. Exactly.
Yeah,
So
that's what they look like. You can find them here. But yeah, they'll, it's kind of like, it's divergent evolution due to being in very different environments.
But they're related.
Yeah, they are related. They are marsupials. Yeah, the genetic differentiation we're talking about is very small compared to the difference between kind of, either marsupials or eutherians, or marsupials or monotremes, for example. And so that kind of just leads, lastly, to humans, just to kind of put a pin on everything. We've now but what this is just highlighting is you can even add, with all the magnifying glasses we've looked at over these past two modules, you can still, even within mammals, kind of zoom in closer and closer till you get to humans. We're talking about the old world and new world monkeys, as well as the great apes, and ultimately humans from there. So that's it. Yeah, that's it. So now we've got about 30 minutes left where I can just leave the floor open.
Exam review.
Yeah, that's what I mean, is leave the floor open to questions. Yeah.
Oh, you're not going to review things.
Well, I want to review whatever y'all want to talk about.
But last time you made a document.
A document.
Like review materials.
Wasn't that just
Like
about
stuff
the
about-
reading?
Well, that was just the reading. Yeah.
The thing that you went over in class?
Yeah, I was just pulling up slides as the questions came. I have slides that I can pull up, so whenever y'all were asking a question last time, I was just looking for the slides on PowerPoint, pulled it up, and then we went over the topic. But yeah, we can,
You said protists will be a big thing to get rid of the protists.
things you want to know are those key features. When we talk about protists, keep in mind that these are the first eukaryotic organisms. So talking about the endosymbiotic theory kind of always gets brought up when we first talk about protists, because it's those first eukaryotic cells. And the eukaryotic cells, you have to explain why they now have membrane-bound organelles, why they have mitochondria and chloroplasts, depending on the cell, and that's where endosymbiotic theory comes in. That's why it gets lumped into the protist, right? The other thing to know is where everything else came from. Which of those branches? That's really all I care about in terms of knowing the purpose of a diatom or something like that, or knowing one specific group and its ecological or economic importance. I kind of tend to leave that for 1102 because there's a lab that's just totally dedicated to that. But here I'd rather kind of not have y'all just memorize for memorization's sake. There's a bigger importance to knowing about those three groups because they led to something bigger, to the plants, to the fungi, to the animals. So those are really the two main things. Know the predecessors of plants, animals, and fungi, and what the traits are that kind of the clues that help us confirm that, and know about the endosymbiotic theory.
So like the cyanobacterium.
What
Oh wait,
about?
that's algae.
Yeah.
Is it the same format as like short answers, multiple choice short answers?
The exam? Yeah, so it'll be, I think, somewhere between 30 and 35 multiple choice questions, followed by those 8 short answer questions, where you have to do the first 4, and then 5 through 8, you choose 3 of those 4.
What should we review with like getting prepared for the free response questions?
For the free response questions? Oh, okay, so the biggest chunk of advice that might be helpful for, I think, maybe a fourth of y'all from what I noticed is, and this is typical each semester, is that timing tends to be our worst enemy. So you can always ask yourself, which would you rather have left to do in the last 2 minutes of the exam? 5 multiple choice questions or 5 short answer questions?
5 multiple choice.
Yeah, multiple choice questions, you typically have a 25% chance of getting them right by random chance. You can at least, in the last second, bubble in A for everything. And you may get some of those points. It's very unlikely that the same would happen with the short answer. If you just randomly throw letters out there, you're unlikely to get a coherent sentence, much less the right answer. So my advice is, even though I can't really change the format of how the exam is presented, because we try to keep everything the same across sections, my advice is do the short answer first. Look at those, and then move on to the multiple choice. I think that's just personally a better strategy to go about it. I just can't myself put the short answer questions first.
So like, what should we read thoroughly and sufficiently? Which chapter should we read very, very thoroughly?
Just look at
So
the
that way,
read-module, right?
so that way we know how to do the free response question.
Just look at the read-module.
Yeah, I mean, I can't guide you to, specifically like, okay, questions 1 through 8 are mostly about animals, right? It's not fair, like, for me to say that when the other sections aren't getting that big of a hint, kind of, yeah. But, that said, if you ask about a topic, I can tell you, what are the parts you want to make sure you have down, right? So, think about, what we try to do with these short answer questions. We try to get a good representation of everything that's been covered in the module. So, if you could divide modules 3 and 4 into, let's say, 5 things, 5 topics, how would you divide modules 3 and 4?
Plants, animals, viruses. What was it, bacteria? What do you
Yeah.
call them, the viruses?
Bacteria, And then, yeah, protists have got to be in there too, right? Plants, animals, fungi are protists. And so you might expect a question, well, okay, if protists have got to be in there, you know it's going to be one of those two things at least. Maybe it'll be both.
So it's not going to be a big critical exam.
Right, well, or if it is, you know, I've only been talking about two aspects of protists, right? Where did we come from? Where did the other things come from? And endosymbiotic theory. So if you have those down, that's already kind of a huge chunk I'm telling you you don't need to worry about for protists.
Wait, why don't we need to worry about it?
Because we've only learned two things about it.
Yeah,
Oh, okay.
yeah. Yeah. So the other things, think about plants. What did I say was kind of the biggest chunk about plants you want to make sure you have down? What did we talk about the most when we talked about plants? Yeah, alternation of generations for sure. What might be another thing?
Diploid, haploid,
Plant phylogeny, seedless, vascular.
Yeah, those four main groups, right? What is the difference between a seedless vascular and a non-vascular plant? The difference between gymnosperm and angiosperm, seed plants versus seedless plants. Yeah, so all that might be more important than knowing the definition of mega gametocyte, right? We didn't really talk too much about that. So if you're trying to spend 5 minutes remembering the vocab term of one small aspect of the alternation of generations, think more, okay, do I understand that diagram? That switch from haploid to diploid, the steps involved there, as opposed to just focusing on those key vocab terms.
Would you say that the module reading guide basically gives a good...
Yeah, the reading guide definitely does. I mean, if you can master the reading guides, for sure, you're in the driver's seat.
Professor?
Yeah.
Sorry to cut you guys off. 25 to 27, are those the three chapters that are going to be consistent?
Honestly, I can't even tell you what chapters y'all read because I go with the...
A different book?
Well, no, I try to explain these concepts in my terms, right? So if there's a concept on alternation of generations, I try to explain it in my terms as another way for you to help you understand. My goal is always to try and kind of apply this, quote unquote shotgun approach. I try to provide the lectures, which some people, works for some people, and for other people, the aspect of me talking constantly puts people to sleep. It's no fault of yours or mine if that's the case. It's just we all learn differently.
I go by the reading guide, look at the pages on the book.
The reading guide is
I look
another
at
resource.
your slides and how you simplified it more, and from there I just get it.
Yeah, and some people, the activities tend to be the most kind of,
Helpful?
well, it seems to be the most divided. People tend to either really like the activities or hate it. And so if you find yourself in one camp, just realize that there's a big part of the other class that actually does, either like them if you hate them or hate them if you like them. For me, the group activities never feel natural to me. That's not how I learn, but it's effective for a big group of students. And so I try to just make everyone aware of these different strategies. Some of them will work for students, some of them won't. It's very unlikely that they all work for anyone. Yeah. So I don't even know who asked whatever question led to this answer.
The chapter sheet.
Sorry.
What was the question? Oh, the chapters. Yeah, so I don't know what chapter.
I think it's 17.
It's
24
the
through
module, it shows
It's
on
in
the
the
reading guide.
syllabus.
Yeah, the reading guide will point to the exact section. Because you'll often see that a chapter is broken up across modules. Like I think we revisited,
We did 17, then we skipped all the way to 24.
17 and 24 was in one section, right? And it was only parts of 24.
Yeah.
We saw the rest of 24 in a different module. So look at those reading guides to be aware of which sections you want to remember. Because relearning Module 1 stuff is going to be useful for the final, but for this exam,
Yeah, because
It's
module
not gonna
3
do
is
anything.
17, 24, 25, and 4 is 26 and 27.
Okay, yeah, there you go. Yeah.
It's going to be a little bit more, it's going to be at 26 and 27.
Does everyone know about the body cavities? This difference between acoelomate, pseudocoelomate, and coelomate. No.
Platyhelminths. Platyhelminths
Yeah.
are ace.
Yeah, so there's really only one group that we tend to look at when we're doing the acoelomate. That's the platyhelminths. They have no body cavity. They're actually super solid all the way through. You'll find one hollow portion in them, but that's their intestine. So everything else has, in some way, a body cavity, some fluid to protect the organs there. So the pseudocoelomate basically have, okay, so platyhelminths have no body cavity. It's tissue all the way through until you're inside the intestine. Pseudocoelomate, what'll happen is you'll kind of cut, and then you'll get into fluid, and then you'll cut another set of tissues. It's the difference between the ectoderm and the endoderm. So there's organs, specifically digestive system, and it's just floating in this fluid cavity. And then there's the outside, the exterior of the organism itself, the ectoderm.
There's no mesoderm?
Not in pseudocoelomates. Sometimes there might be.
But I thought
We'll
the
talk about it.
mesoderm is where the viscera came from.
So in truly coelomate animals, that endoderm is suspended by not just a fluid, but by ligaments and structures, tissue that connects to that ectoderm. It's kind of suspending everything by muscles as well. And that's the mesoderm and its primary function.
Okay.
And so what's that?
And pseudocoelomates do not have a mesoderm.
No, so they do have, they can have a mesoderm. So this distinction of whether an animal has a mesoderm or not gets to a different term that has to do with blastic. Anyone remember these vocab terms? What's that? Diplo and triploblastic. So diploblastics don't have a mesoderm. Triploblastic individuals do.
And triploblastic
Chordata.
to the proletarians, or the bilateralists, I'm not sure.
Yeah,
Mostly,
so
right? And then the diploblasts are braiding.
yeah, so jellyfish are essentially diploblastic. So what we can, one thing that tends to trick students is that endoderm, ectoderm, mesoderm, what does it all become? One way to remember this, you don't necessarily have to memorize all the things it becomes, so long as you kind of think about the logical things that kind of the big things that they develop into. So the endoderm develops into a few different systems, but the main one is at kind of all of our centers. So even in the simplest organism, the simplest animal, it's got to eat, right? It has to ingest, travels through the body, and goes out. That digestive system is one of the primary systems that the endoderm becomes. It's kind of what's at the center of most organisms, is that digestive system. Everything else kind of grows around it. And so, there are other things that the endoderm will become, like a circulatory, part of the respiratory system. But the main one, the key one that might be the easiest to remember is if you see digestive system, think endoderm. Because even, your textbook always shows it in that super simple diagram of, okay, we're cutting some hypothetical worm, some very simple organism where we have, if we're talking triploblastic, we've got kind of three parts. We've got a mesoderm. This is actually, I think, what your textbook shows. We've got, and then the last here. The endoderm. So at the center of all organisms, we're kind of getting at that digestive system. So think that for the endoderm. This, all those cells that make up endoderm. I know that's super tiny, but endoderm, the most interior. And what's at the most interior of every individual is that digestive system. All the other systems kind of grow around it. What holds these systems together? The mesoderm. And so what's kind of, if you had to think of one massive set of tissue that's between our digestive system, our most outside layer?
Mesoderm.
Muscle, right?
Oh.
Yeah, musculature. So when you think of mesoderm, think muscle, M. Think of M&M or the green M&M. Mesoderm becomes the muscles, most musculature. So mesoderm. And then lastly, the ectoderm, our most exterior, our skin. Yeah, so, yeah, the epidermis, ectoderm, so ectoderm, think of skin. You might see a question pop up where it's kind of showing all these other organs too, but really look for those key ones. You know, if you see, okay, these type of cells become, you know, the lungs and, well, let's say the heart, muscles, red blood cells. Well, you saw muscles in there, right? So I know that's got to be the mesoderm. I maybe don't remember that it also produces red blood cells, but I know that it is related to the muscles. The same goes for, you won't get the question the other way around. You know, what does the mesoderm not become? And 3 options are right and 1 is false. Really, if you kind of know the big ones, you should be good in this sense. And, you know, just by the fact that it's got these 3 dermal cells, 3 means tryptoblastic. If it's lacking any one of these, it's the mesoderm, and that's the diploblastic.
Professor, I have a hard time compartmentalizing and differentiating between this and blastula, blastospore, and gastrulation and all.
Yeah. So, I mean, part of the terms come to build off of each other, right? The triploblastic, part of it is getting into, you know, this idea that the digestive system really is the center of everything. When we talk about the blastopore, whether it forms the mouth or the anus, what the blastopore is doing is slowly forming a digestive system. So, you know, if you look at the diagrams in your textbook, you'll, you know, you start off with the cell at the earliest stage. It starts dividing, and one thing that starts to occur is, you know, you get sort of this first indentation. Start to get bigger and bigger, right? So this indentation grows and grows until eventually it kind of connects on the other side. You have what eventually becomes the formation of what will become that digestive system. And so that's why it's partially related to diploblasty and triploblasty, because at the center of those organisms is that digestive system. And the blastopore is the formation of the digestive system. And so what it becomes, you know, how it starts differentiates between those protostomes and the deuterostomes. So if this is the side that's going to become the mouth, what do we call this? Protostome. And if it were the anus, the deuterostome. Yeah. Is the process? Yeah, that is occurring. So we'll talk more about it in Module 5. How we'll see. see gastro appear again, but gastrulation, yeah, relates to digestion. Just comes from the same word as gastronomy, cooking. So it's all about that digestion. And I'll answer more questions, but as y'all are thinking of them, or as we're going over them, I do want to play one video that's just really cool. There was, if I find it, I'll post it on Blackboard. But researchers recently kind of put a live webcam in a petri dish grown organism that I'm blanking on which one it exactly was. I can't remember if it was a seahorse or a zebrafish or something else. But you see that blastopore formation happening in real time, like it's accelerated. So not real time, but in fast forward time. Other questions? Yeah. Keep them coming.
So wait, the mesoderm is where the viscera come from? Or no, that's the muscles.
Yeah, the musculature.
What does the endoderm do?
The endoderm is the formation of that digestive system.
Oh, the endoderm is the digestive system.
Yeah, endo, the most interior. Endo meaning within, right? Yeah.
Can you go over the transduction and transformation?
Yeah, but first, Jelani.
Do you recommend going over the C-rat that we've been doing?
Oh, like the IRATs and.
IRATs.
Yeah, for sure.
Yeah.
Yeah, transduction and transformation. Yeah, so all ways of genetically transferring information among bacteria. So those are just two. There are other ways, binary fission, which kind of separates from the other three. Binary fission, the reason it separates is because we refer to that as vertical gene transfer. It's the creation of a next generation of bacterial cells. The other three, transduction, transformation, and conjugation, are horizontal genetic transmission. It's transferring genetic material to a member of the same generation.
Wait, you said transduction, transformation,
And conjugation. So conjugation is that one where the mating bridge forms and the bacteria will transfer what's called an F plasmid, a small part of its DNA that is specifically meant to be transferred from one bacteria to another. It's never mutual exchange. It's always one-way transfer. And it's the transfer of an F plasmid, a designated plasmid DNA, to the next individual. Transduction and transformation is where sort of outside elements come in. One of them involves viruses, kind of relying on bacteriophages, which will kind of remember how viruses are made they replicate by essentially hot wiring the host's own tools. It'll use the host tools to make all the pieces needed to make a virus, which include more virus RNA or DNA, the capsomeres that make the capsid, and all the other pieces of the virus, any necessary proteins or ribosomes. The host makes all of that for the virus. And so what happens is, as you're assembling those pieces, sometimes mistakes happen. You get accidentally grabbed from the wrong Lego set, and you grab the bacterial DNA and put it in that virus, and now you've created a virus that has bacterial DNA also in it, and it'll transfer to the next bacteria that it takes over, that it infects. So is that one transduction or transformation?
I'm playing numbers again.
Yeah.
Oh, is it? Okay.
Yeah, so transduction is going to be the virus transfers genetic material. Transformation, it's a little bit different. It just doesn't involve a virus necessarily. So sometimes bacteria can find themselves in strange environments where, you know, everything sheds, right? So as things shed, DNA can be found anywhere. Just like, you know, if you're a CSI forensics person trying to find DNA of some culprit, you'll see there's DNA on any surface, right? So there's bacteria on every surface too. So sometimes bacteria come across foreign DNA, and that foreign DNA just kind of makes its way into the bacterial cell. Because bacterial cells, just like all cells, aren't entirely impermeable. Things go in and go out all the time. And sometimes it'll recognize DNA and it'll just find a part of that genetic sequence that triggers a receptor to allow it in. That gets incorporated. Now there's, you know, once you're inside of a bacterial cell, all the proteins assume, hey, all the DNA that's here belongs to this cell. So it gets incorporated eventually into the genome, and now the bacteria has foreign DNA as part of its genome. This happens predominantly in bacteria, but sometimes there are cases of it happening in other organisms too. Beetle genes have been found in trees, in tree genomes. So vertical genetic, or this horizontal genetic transmission, which we call transformation, doesn't only happen in bacteria, but it happens in them all the time. For other instances, it's super rare. Yeah. All right, we're at 4:15. Yeah, remember that, you know, I'll just like we did with exam 1, right before exam 2, I'll give the opportunity to answer any questions. So take this weekend to really study up, note things that you're just not getting, and we'll try and tackle them. We'll have like 10 to 15 minutes before the exam to do it. Remember that, you know, if there are no questions, you can start you'll have more time for the exam, but you know, most people were able to finish pretty, you know, with plenty of time, but yeah, just keep that in mind.
Thank you.